So I do want to start with a couple of caveats. The first is that my natural metabolic pace is quite fast. I tend to accelerate as I go sometimes. I'll try not to, but it does happen. And so if you have questions, just interrupt me. I'd much prefer to take questions as there are points that need clarification rather than waiting to the end. As Emily mentioned, I'm part of the HMBA, which is the Human and Mammalian Brain Atlas project led by Ed Lean at the Allen Institute. We also have our own UO1 that's a no-cost extension. A lot of the stuff I'm going to show comes from that. And then there's this new NIH Brain Connects initiative that we have a UO1, and Anastasia Yandiki has a UO1 that we're part of. And so I'm going to give, so I have 2 talks listed here. It's really not 2 talks. It's really like half a talk and 3/4 of a talk, something like that. But I can go fast enough that it fits in one talk. And so this stuff is going to be kind of background for the second part. And the other thing I'll say is that when I was talking with Kiku and Emily about what to present, they wanted to make sure that I knew that this was for a diverse audience, and including a lay audience, and that's fine. And so the talk is kind of geared for that. And so I'm not going to go into a lot of technical detail, but I'm happy to. Like, if anyone's interested in model architecture or whatever, I'm happy to talk about it at any level that you want. And I'll try to show people as I go. This is work from a ton of people over a lot of years. And these are the kind of the primary ones, but I'll point people out as I go. Okay, so there's some issues with trying to use modern deep learning, particularly in the world of MRI, clinical MRI. And it's kind of easy to come up with this kind of list. We need a lot of manually annotated data, like 2 or 3 data sets is not enough. 5 or 10 is probably not enough. You can do a lot with 5 or 10, but if you have 1,000, you can do a lot more. Annotation quality varies a ton. If anyone here has ever tried to label 3D data sets, it's an awful thing to do. It requires years of therapy for the rest of your life after you've done it for a while, because we as human beings are terrible at this. Like, we have no phenomenological we have no reason to have any competency at this, at taking 2D sections and stacking them together in our head in 3D. And if you've done this, what you do is you label one slice and you look at it and you're like, oh my God, that's perfect, that's brilliant. And then you label the next slice, you're like, that one is a work of art. And then you flip it into the different orientation and it looks awful. Like, there's no consistency from slice to slice. And so this is just a hard thing to do, to generate ground truth data. This one is a big problem for MRI in particular. That is, if you train on a particular type of data, you will always be biased towards it. So if I change the way my acquisition looks a little bit, then the network accuracy will go down. And so if all my Alzheimer's are scanned with one type and all my controls are another, am I finding an effect, or am I just finding the fact that the images are different? Even if we're really good at annotation, some stuff is really hard to annotate, like 6-slice MRI. If I have 7mm slices and the spacing of my cortical folds is 2 millimeters, that's hard to do. And then lastly, even if the data quality is high, there are some things that are just intrinsically difficult. We see like the growth of lesions over time. And so you have a voxel that's maybe a little bit brighter on time 1 than it should be, and on time 2, maybe it's a tiny bit brighter than it was on time 1. Is it part of the lesion? Like, these boundaries are kind of fuzzy and vague, and so we're not very good at annotating them. And I would argue that this has been a problem for us clinically. It has been the primary barrier to our ability to distribute things in the clinic and to have radiologists get automated measures of hippocampal volume. And to me, this is a failure of capitalism in medicine, because in radiology, people pay for scan slots, not for time. Like at Martinos, we pay by the hour. But if you go and get your brain scanned, you will pay for an MRI scan. And if that MRI scan is 5 minutes or an hour, it's the same price. And in MRI, like, MRI is not like photography. The process of acquisition is kind of accumulating information over time. And so in an hour, we can acquire a much better image than we can in 5 minutes. But if we acquire a much better image, now the radiologist has to read 5 times as much data. And that's also a financial hit for the hospital. And so there are these incentives, there are these capitalist incentives for crappy data in the hospitals. And we fully meet that bar and routinely acquire crappy data. And so the question is, what can we do with it? Are there things that we can do with the crappy data now with the advent of deep learning that previously we couldn't? Okay, and so this part of the talk, I'll talk about various synthesis-based deep learning. I'll explain what I mean by that. I think the biggest challenge in the development of this stuff is this slide that I stole from Anastasia Yandiki, which is that it takes so long to train these things. you know, you have an idea and you're like, oh, this is going to be great. Then you set it training, and then 2 weeks later it's converged, and you run it, and you're like, oh my God, I did something stupid. And then you fix it and you start it again, and then 2 weeks later, you know, it repeats. And so the development cycle has really slowed down because of this. Okay, so what do I mean by the synthesis stuff, and why do we need it? So MR is a wildly flexible technology. If you don't know much about MR, the reason it's so popular is that we can derive contrast from many, many, many different things in the anatomy from it. So here's an example of what we'll call MR modality. So T1 weighted, white matters bright, gray matters dark. T2 weighted, you get the inverse contrast. Proton density weighted is kind of somewhere in the middle. This is a sequence commonly used at MGH for deep brain stimulation targeting. You have the flexibility to null certain tissue classes. So we want a technique that works for all of this stuff. But in MR, we can do a lot of other things. Even within the class of T1 unweighted images, there are a lot of other image types that we would all call T1 unweighted, but they have different contrast properties. The gray matter is darker, it's brighter. In MR, we frequently play the game of sacrificing SNR for CNR. That is, we trade off increased noise for increased contrast. And so this, for example, is MP RAGE, which has higher contrast to noise than a flash scan, but lower SNR than a flash scan. And then we can mess around with the resolution, right? We can change the orientation of our slices. So this is an axial acquisition, a coronal, a sagittal one. We can skip space in the slice. So we can take a 3 millimeter slice, skip a millimeter, take a 3 millimeter slice, or we can just take thick slices. And so we would like a technique that works for all of this stuff, and that's hard. So one approach is just to build a supervised CNN. These days, that usually means a UNet. The UNets pretty much outperform anything on at least image analysis stuff. So I can take a supervised CNN, take this type of input, manually label a bunch of outputs, and I could create a nice CNN or a UNet that can automatically label axial T1 scans, right? And that works if I can, manually label 20 or 50 or 70 of these. But it falls apart if I apply it to something else. So if I apply this axial scan to a coronal scan, this is the result. Like everything just falls to pieces. And of course, it even gets worse if I apply it to a different contrast, like to a T2 weighted scan, okay? So what's the alternative? Well, sorry. And so this is the case across kind of broad changes in contrast. But even for small changes in contrast, networks are super good at picking up tiny details in the images. They're great at cheating. If you haven't ever trained a network, networks are the best cheaters you will ever encounter. And so you have to watch them like a hawk, because they will learn a way to solve the problem that is not the way that you want it to solve it. And so this is an example. This is hippocampal volume compared to ground truth. So this is a dice score. So 1 means perfect overlap between the ground truth and the automated, and 0 means no overlap. And this was kind of our state of the art before the advent of deep learning. And if you train a UNet on just the T1 weighted images, just this class of T1 weighted images, these MP RAGEs, you can do pretty well. You can get 88, 89, close to 90% accuracy. And if I take this network and I apply it to a flash scan, which is a T1 weighted scan, like the white matter is bright, the gray matter is intermediate, and the CSF is dark, you get this catastrophic loss of accuracy. And it's because the network has learned these subtle details of what it means to be an MP RAGE, these frequency filters that we implicitly apply on them, and it doesn't generalize. And so this we generally call the domain gap. That is, a network trained for this sequence doesn't work well for this sequence. And this is a little bit of a toy example. There are things that I can do to make this better. I can augment my input data, I can add noise, I can mess around with the contrast, and that will help. But I think you will never get away from the bias of the MP RAGE sequence, because that's what you're trained on. And even worse, you will never know if you got away from the bias, because we can't ever prove a lack of bias, right? Proving a lack of bias would be proving the null. And so all we can show is that there's no bias at a certain level. Okay, so what can we do about this? We'll skip this. So we could manually label every single type of scan under the sun, but that's, of course, not tractable. Okay, so the proposed solution is something, this was actually Adrian Dalka's idea, and it came from this process of trying to augment the data. And augmentation in deep learning means taking your training data and making it worse in various ways. Adding noise, spatially warping it, adding shading, intensity artifacts over space, this kind of thing. And at some point, after kind of making these augmentations increasingly wacky, Adrian had the insight of, why do we need the images at all? And so the way we train networks these days is without any real data. So the way it goes is we start with the 3D anatomical segmentation like this. The segmentation doesn't have to be manual segmentation. It doesn't even have to be that accurate because we're not going to pair it with image data. Then from each one of these, we'll sample a spatial transform to create, a subject. So we'll warp it in some way. So here are a bunch of different subjects, synthetic subjects warped. And then for each of these tissue classes, we will sample a set of intensities. So I'll sample a mean and a variance for the white matter, a mean and a variance for the thalamus and for the basal ganglia, and I'll just make up an image, right? And this is what the images look like. And the images are crazy. And Benjamin Biot, who I'll show in a second, who wrote, here, let's go to that. Here's Benjamin. Benjamin was Eugenio Iglesias' grad student when he wrote this stuff. He's now a postdoc across the street with Paulina Golan at MIT. And Benjamin had trouble publishing this. And the reason he had trouble publishing this is people looked at these images and they kept saying, well, that doesn't look like an MRI. Your MRI simulator is awful. What are you up to? And for some reason, we can never explain to people sufficiently that we're not trying to simulate a realistic MRI. All we're trying to do is kind of span the range of variability we expect to see in the input sequences. And the idea is that if this data is kind of more variable, more difficult to segment than the real data, that the network, by the time you get the real data, will be like, this is easy. And I'll show you kind of some more quantitative analysis than my head swipe. This is easy. But it turns out that that's the case, that that in the cases where we took this data and we tried to make it more realistic, we found accuracy went down. And that we're better off kind of spanning this range of wide variability than we are trying to make things realistic. And so this works. So here's a set of images that were all segmented by the same UNet. The UNet was trained without ever being exposed to a real MRI, just all these synthetic data sets that we created. And here's, decreasing resolution, increasing voxel size, so 1 millimeter to 7 millimeters, and then 2 different types of scans. And yet you get pretty much the same segmentation. So it's pretty much invariant to the input data. And of course, if you have 7 millimeter slices, you're not going to extract as sensitive information as you are from the 1 millimeter data, but we're not biased towards one or the other. And this is just to show that point, that here's the data set that it was trained on, and when you train a UNet on that data, it does great. But this thing shown in green is the synth-seg stuff, and you see it maintains high accuracy across every different type of MR sequence, and you can even give it a CT scan, and it'll do something reasonable, even though the CT shows no contrast between gray and white. Okay, so that's one game we can play. Another is something that Eugenio was up to, which is synthesis. So I'll skip this. And this is kind of important for some of the BICAN stuff, which I'll explain why in a second. And so the idea here is that we're going to do the same thing. We're going to start with a pool of label maps. We'll sample a label map. We'll warp it to create an individual subject. But now, instead of throwing out the image intensities, I'll use the image intensities as targets. So this is the real scan that was acquired for this subject. It's deformed in the same way as this one. So these intensities match that. Then I'll take this scan, and I'll do all sorts of awful things to it. I'll blur it. I'll create thick slices. I'll put maybe some subject motion into it. I'll add noise. I'll change the contrast so it doesn't look right. And then we'll do regression. And so the game we play is this is input to the network, and this is the target output. Right? And so we'll try and synthesize what a high-resolution 3D 3T scan would look like from any input data. And this works pretty well. So here's an example of 3 different input scans. There's a 6 millimeter flare, there's a 5 millimeter sagittal turbospin echo, and a 4 millimeter coronal T2. And we can turn them all into something that looks like a 1 millimeter MP range that we can then kind of put through any anatomical analysis package world. And in BICAN, specifically, we've been applying this to these low field scans we've been getting from a hyperfine scanner. So this is part of Edlene's one, and Christine McDonald and Dirk Keen bought and cited a low field scanner, a hyperfine scanner in the autopsy suite, in Dirk's autopsy suite. And so every brain that now comes to autopsy gets wheeled into the scanner and gets this type of low resolution T2 weighted, because that's all we can get on the low field scanners, 3 millimeter isotropic. But with this technique, we can take this 3 millimeter low field scan and turn it into a 3T MP range. And further, we can take this 3T MP range and stuff it through FreeSurfer and segment, the cerebellum and motor cortex and whatever. We can get these kind of detailed anatomical parcellations that are registered to this autopsy sample. And this is Oh, that was a slow transition. This is what I was chatting with Kiku a little bit at lunch. This is Kartik's work. So Dirk will take photos of the slabs after dissection. But before dissection, we run a surface scanner over the brain, and we get this type of 3D mesh. And it only gets a millimeter or two into the folds. It doesn't get into the depths of the folds because we can't see it from the exterior. But it provides us with enough geometric fiducials that we can take these 3D slices, or sorry, these 2D photographs of these 2D slices and stack them up into a 3D volume and use that to register it to the hyperfine scan. And now we have a path to go from any in vivo atlas to the neural path. Right? And so that's what's shown here. We've taken, I guess this is our standard cortical parcellation, and projected it onto the neural path. And so now we can say to Dirk, we want you to take out a slab that's in motor cortex, or maybe it's in the default mode network, maybe it's the frontal component of the default mode network. We can bring kind of the full power of in vivo imaging to guide the neural path. And of course, we can go in the other direction too once we have the neural path results. Any questions on that stuff before I continue? Okay. Another thing we can do is we can detect change. So change detection is, if you talk to a neuroradiologist, they will tell you that they spend the majority of their time trying to detect change in the brain. That is, they'll look at 2 scans and they'll try and find the same slice. And they'll say, has anything changed? Has the lesion grown? Are the ventricles bigger? Is the hippocampus smaller? Because these are the biomarkers for things that are going on in the brain. And so hospital information systems, so if we do a research study, we are very careful to prescribe every single thing that we can think of to stay the same over the course of the study. We'll use the same scanner, we'll use the same head coil, we'll use the same receive coil, we'll use the same sequence, we'll try and position the subject to the same place in the scanner. That's all out the window when you get a clinical scan. In the clinical scan, you will not even be on the same scanner. Like there's no provision in the hospital information systems to say, I want this to be a 3T Siemens scan with this head coil. So your time 1 scan may be a GE 1.5T and your time 2 is 3T Siemens. And the result is that if you compare the images, there will be huge differences between them. And the vast majority of those differences are technological in nature. They have nothing to do with the brain. They're just the fact that the way we acquired the data was different. And so we've been looking at trying to synthesize our way out of this problem because change is a very difficult thing to label in the brain, like what real anatomical change is. And so here what we do is we take a time 1 scan in the same way we did before, a time 1 label map, the same way we did before. We'll synthesize the time 2 scan from it, like we'll move it in space a little bit, and then we'll introduce a little bit of atrophy somewhere in the brain. And because we know exactly where we introduce the atrophy, we have ground truth for the training. And further, we can introduce all sorts of intensity artifacts. We can add noise, we can make the contrast different, we can add bias fields, blur the data. And then the question for the network is, can it take the input pair and find the true anatomical change in the presence of this kind of sea of technological artifact? I should note this is work with Sean Young. And this also seems to work pretty well. So these are real scans. This is a multiple sclerosis, a subject with MS, and here's a subject with Alzheimer's disease. And if I take these 2 scans, so time 1 and time 2, this is real data, and I rigidly align them, and I just do a pixel-wise difference, like I look at just the absolute value of the difference in the pixel intensities, you'll get stuff everywhere. So you get all this stuff outside, and this has to do with fat shift in MR. But then you get all this stuff kind of in the brain as well. And this stuff is not real anatomical change. There's nothing going on, out here in whatever the bone of the skull. This is just the fact that the MR scan is different. But the network does a nice job at finding kind of these ventricular changes and also this growth in some white matter hyperintensity, and the same thing with the MS lesion. Okay, almost done with the synthesis stuff. So this is some work that was done, and this stuff I'm going to show in the second part of the talk also. So this stuff pay a little bit of attention to. This is work from Adrian Dalka. So Adrian's PhD work was on doing nonlinear registration with deep networks. You call voxelmorph. This is also across the street. Adrian is now faculty at MGH, and he and Malta developed this technique called synthmorph. And in synthmorph, we have these 2 synthetic images, and they're from the same subject, and we can distort them a little bit. So there's some nonlinear distortion. Sorry, they're not necessarily from the same subject. So there's a nonlinear warp that relates them, and we would like to recover that nonlinear warp. The problem here is that the image intensities are somewhat random. And there aren't really good cross-modal similarity metrics in kind of the image registration world that work well for nonlinear registration. They work pretty well for rigid and linear, but it's a little bit ill-posed to try and recover a nonlinear warp in the presence of a nonlinear change in the intensities. And so what Adrian and Malta observed is that in the synthesis framework, we don't have to compare these images. We don't have to say what transformation maximizes the similarity between this image pair, because each one of these images comes with an associated label map. And so instead of comparing images, what we could do is compare label maps and say, I want to recover a nonlinear deformation that aligns hippocampus with hippocampus and amygdala with amygdala and thalamus with thalamus. And I don't care about the images. The network's job is to figure out what that means in terms of image intensities. And this works quite well. And further, so I'll come back to this in a second, we don't even need brain images. We can make up the input label maps. So we can make up images like this. This is kind of multiscale Perlin noise. So the network can we can take this image and then warp it and then train a network to recover the deformation between them, because we have both the labels that give rise to this image and the image itself. And this will be important because sometimes what we want to register is not a brain. And we did a parametric study on how well this works. So this is the target of a deformation shown in the bottom row. This is the source of a deformation shown in the top row. This was a scan that we have a forward model for image formation. And so we can actually synthesize images with different contrast. This is a T1 weighted image, and this is a fully PD weighted image. And now I'm going to run image registration on each of these pairs and look at the accuracy of the resulting alignment. Okay? So left to right is kind of more and more cross-modal. Like this pair is the same contrast. This pair is completely different contrast. And then these are different image registration techniques. And the blue one, the top blue one, is the synth-morph that was trained on brains. And the second blue one is the synth-morph that was trained only on shapes. So it's never seen a real brain image. And what you can see is that the accuracy is pretty flat. Even out here, where we lose contrast, where the gray-white contrast is largely gone, the accuracy stays pretty high. And so we can generate nonlinear registrations kind of between any pairs of brains and further between any pairs of shapes. And I'll come back to that in a little bit. Okay, that's the end of the synthetic deep learning stuff. Any questions on any of that? Okay, on to multiscale stuff. So this is really the kind of core of our contributions to BICAN and Brain Connects. And in the end, I think probably like a lot of people here, we want to construct a multiscale atlas of the human brain. And to do that, one of the things we were discussing at lunch is the crazy array of spatial scales over which structures of interest occur in the human brain, from nanometer to multiple centimeters, right? Ion channels to cortical fold loop patterns. And we want to account for all of these things. So we would like to build an array of image acquisition types that reveal these properties of interest, and that could be genomic or proteomic or transcriptional or functional or anatomical or connectional. We want to register things, so we want to build registration tools that accurately align these different image types across scales and contrast. And it's worth noting that this has to be cross contrast. There's a reason we're doing the imaging, that we're doing a lectin stain, for example, because we care about the vasculature, or we're doing a new N stain because we care about the neurons. And so by construction, our images show different things. And so the contrast is inevitably going to be different across these registration targets. Once we have the registration, we want to build individual subject level modeling tools so that we can situate the results of whatever we're acquiring into the context. So what cortical layer is it in? What column is it in? What cortical area? This kind of thing. And then finally, once I have all this data at the individual subject level, how do I put it all together into an atlas and register my different subjects together to build an atlas that is representative of these properties that I've just observed in this unique data set. And the thing that's hard about this is that we want a tool that can register stuff that it doesn't see. And what I mean by that is, you can imagine a BICAN study where we, do a complete transcriptional characterization of, the substantia nigra. This is Emily's project. And we find different cell types in the nigra, right? And now we have another subject, and we want to register them such that those populations of cells align across subjects. Well, that's fine for ex vivo studies in which we've done this transcriptional analysis. But what if I want to do it in vivo? What if I want to apply it to Parkinson's patients? Then I want a technique that aligns these areas without being able to see them. Right? We want to be able to align things that we can only see during training and not necessarily at inference time. And I'll talk about how we do that in a little bit. Okay, and as I mentioned, this is part of this UL1 that Hui Wang and I have at MGH, and David Boaz at BU, Patrick Hoff at Mount Sinai, and Francesco Pavoni in Florence. And so in this project requiring ex vivo MRI, we acquire optical coherence tomography, which I'll describe in a little bit, because probably people aren't familiar with it. We were doing it at BU in the previous grant. In the Connects instantiation, Hui is building a rig at MGH for it. Then we do light sheet microscopy, so fluorescent light sheet microscopy at Florent. There's some stereology that goes on, some deep learning to segment cells, and then we want to put them into a coordinate system where we can start to make statements about human variation, like how many neurons are in this layer in Broca's area, for example. And then, as I mentioned before, we're also part of this HMBA grant, the Human and Mammalian Brain Atlas that Ed Lane runs at the Allen Institute. And this is where we're doing the low field scanning. And of course, this stuff is of interest because, Steve is here. There's a grant here looking at similar things. And then Casey Chung, who's across the street, is any day now going to get funded also for a UM1 to start looking at proteomics and generate micron scale images of entire human brains. Okay, so in general, in neuroimaging, we have been historically kind of left with this choice of doing coarse images on lots of subjects on the whole brain, or doing unbelievably fine scale images on a few samples in tiny little bits of brain. And we haven't been able to put them together. And so this kind of goes from EM to things like light sheet fluorescent microscopy and OCT and 2-photon imaging. We have built this program at MGH where we use our MR scanners as incredibly expensive microscopes. So we have a 7T scanner, and we use it pretty much every weekend. That's probably an exaggeration. We use it many weekends where we stick a brain in the scanner on Friday afternoon, and then Monday morning we come and take it out. And this is, again, because in MR, time is SNR. And if I want to get to small voxels, I need to scan for a long time. And by long, I typically mean 70 or 80 hours. And so we'll put a brain in, and we'll get, this is a 100 micron scan we got with Brian Edlow a few years ago. We'll get something on the order of 100 micron isotropic across the whole brain. And this ends up being, and actually in the OCT as well, end up being kind of our intermediate spaces that we use to facilitate registration. Because it's really hard to take a 2D light sheet data set and register it to an in vivo atlas. I need some interim targets for that registration. And that's what the OCT and the ex vivo MR represent. So this is an example of the MR we've been doing. This is Divya Varadarajan's work. When we were starting this, we kind of thought 200 micron isotropic was about where we could get. And Divya just totally insisted that was not true. She was like, we can do 120. And the difference between 200 and 120 in an SNR sense is like a factor of, 4 in SNR. I'd have to calculate it. And you lose SNR really rapidly in MR as you increase voxel volume, or decrease voxel volume, rather. But Divya was right. And if, in particular, she came up with a set of techniques that allowed us to measure these tiny little distortions you get in samples. So MR is a magnetic image acquisition technique. And so if you have something that's magnetic in the field of view, it will distort the coordinate system of the image. And in particular, iron deposition in the tissue causes these micro distortions almost everywhere. And so mostly people don't care because your distortion is like 200-300 microns and your voxels are 2 millimeters. So who cares about a 200 micron distortion? And so Divya had to invent this set of techniques to measure and remove these distortions. And for us, it makes a big difference. So this is the cerebellum of a language using, so I'm not allowed to use her name, of a language using great ape that died of natural causes, who we scanned a few years ago, whose name you probably know if you've seen any of the television series on them. And you can see the impact of the distortion correction. All of a sudden we can see 3 layers in the cerebellum where it was really difficult to before. And the same thing is true in the cortex. We lose, I'll talk about vessels a lot in a little bit, but we lose a lot of the vessels if we don't do the distortion correction, right? You can kind of see them, but you can see how they're blurred, but they get much sharper after the distortion correction. And the same thing is true of cortical layers. Here's a band of Baillargeau that we mostly don't see in the distortion. I guess this is probably Sri of Gennari in V1, maybe. But you can see how much clearer it gets after we correct this 200 or so micron distortion. We've also been pursuing this technique called optical coherence tomography, which most of you probably have not heard of. And this is stuff that David and I started way back when. Caroline came as a postdoc, and she's now faculty. Hui also came as a postdoc, and she's now faculty. I showed these slides to Caroline, and she's like, why are you showing 15-year-old slides? In part because I had it, but in part because they're just beautiful. Like this is an image of the human medulla with OCT. So the background image, I think, is like, I guess it's 4 micron pixel size for the whole thing. But if I zoom in, we could also require higher ones where like 0.5 micron. And so we're really almost at the diffraction limit here. And the nice thing about OCT is that in contrast to 2-photon in OCT, we acquire an entire line at a time. So we acquire an entire line in depth. It's not a point-wise measure. And so it's much faster than 2-photon. And so that lets us do these large field of views. And I'll show you what I mean. So here's OCT of the medial temporal lobe. And there's a little inset here, which I'll zoom in on. So you can see kind of the astonishing resolution we get with the large field of view of these mossy fibers in the hippocampus. And then the other game we can play with OCT is we can put polarization on the incoming light. And the reason that's important is that the polarization state of the return light will tell you something about the birefringent properties of the thing you imaged. And myelin is birefringent. And so by putting polarization on the incoming light, we can recover the optic axis of the thing we're imaging. That is the orientation of the axon that we're imaging. That's what's shown here. This movie is playing. So there are two critical things about the OCT. One is this polarization. So polarization sensitive OCT lets us directly image axonal orientation. And the reason this is critical, particularly in regions like the human brainstem, if you have tightly packed fibers, even if you can resolve an individual axon, if it was sitting by itself, with no friends and crying softly at lunch or something, you can't image it if it's in a densely packed region because it just looks all white. Right? To image it, you would need the resolution to see the myelin sheets if you wanted to accurately label individual axons. And that's, an order of magnitude more resolution. That's probably sub, 100 nanometer or so. And I'll show a little bit of that later on. But OCT doesn't suffer from that problem because OCT directly images orientation. So even in tightly packed regions, we get an accurate measure of the orientation of the axons. Okay? The other important thing about OCT, let me play this movie again, is that in OCT, we're imaging a cut block of tissue, potentially whole hemisphere, but we haven't ever built a rig to do something that big. But the imaging is with laser light that penetrates the cut surface. So it penetrates about 200, 300 microns under the cut surface. So this is like formalin fixed tissue. And so the game we'll play is we'll acquire a line in depth of a couple hundred microns, and then we'll cut off the top layer and we'll acquire another line. And this, of course, is the reverse of what you do in standard microscopy, where you cut and then image. And so we're imaging and then cutting, as opposed to cutting and then imaging. And, image then cut then image, it doesn't sound like a big deal. But in fact, it's everything. And the reason it's everything is that we're imaging before we introduce slice-specific distortions. And so to generate a 3D volume of OCT, all we do is stack up the optical images. And that's what's shown here. I'll play this movie again. So here's a little spinning movie of the OCT. And you can see that you can't see the imaging plane. We can't tell what orientation we acquired the data at. And these are 3 orthogonal views through the same tissue. So this is the plane of sectioning, XY, or the optical plane. So this is what we would call an en-face image. But if I take this and flip it 90 degrees, or a different 90 degrees, you can't see the slices. And this is because there is no distortion because we're imaging before we cut. And the physical distortions are really hard to remove. And the fact that the OCT doesn't have these physical distortions means we can use it as kind of a veridical coordinate system. We can take things like light sheet data, and we know exactly what distortions were induced because we have an image of the tissue at micron scale that doesn't have those distortions. Okay. Any questions on the OCT stuff before I continue? Or on anything else, for that matter? Okay. We're also doing a bunch of light sheet imaging, and this is stuff that goes on in Florence with Francesco and Irene, and Danila and Josephine and Beatrice. So here we've been playing with this lipophilic dye called dye D, and dye D likes myelin environments, and it will bind to myelinated fibers, or even very lightly myelinated fibers. And the result is that we can get images of axons with light sheet fluorescent microscopy. And so for the CONNEX projects, we're doing dye D, that's shown here, but we're also doing maybe NuN. I don't know what your experience is with NuN. For us, NuN is really finicky. There are samples that it just doesn't work on for reasons we don't really understand. And so there are other stains that we're still playing with for neuron stains. And then finally, at the highest resolution, the lens group is working on 2-photon after expansion microscopy. And so on the left, I'm showing what we would very much like to do is do the 2-photon imaging on the tissue that we already did the light sheet imaging on. It's not completely clear that's going to work. They've been working on it, and they've gotten to about 3.5x expansion, and that looks pretty good. This is what 4x expansion would look like. And so here you can see myelin sheaths, right? So here we can actually see individual axons at the scale that we can actually label them, right? And so this is kind of our ground truth. And this we're not going to get, we'll get this in like millimeter cubes of tissue. We're not going to get this in centimeters. But the light sheet we'll get over the entire human brainstem. Okay. I'm going to talk about registration stuff, if there's nothing on the acquisition. Okay. So for no particular reason, I'm going to go from kind of biggest scale to smallest scale. So part of the HMBA project is getting human hospice patients to consent to being scanned prior to death and then to brain donation. And there are an unbelievable array of basic questions in neuroscience we have been unable to answer because we've never had this data. So for example, Nancy Kamisher, whose lab is across the street in Braden Cog Sci, has been pursuing human functionally defined cortical areas for decades. She has a spot in the inferior temporal lobe called the fusiform face area. There are various other ones, but in the end, is it a cortical area? Is the face area a cortical area? We don't know. Does it have a different connectivity profile than stuff around it? Does it have different molecular properties? Are the cells different there than in neighboring cortex? Do we see some boundary? And the answer is we just don't know. And it's because we've never had this paired data. And so one of the things we've been working on are tools to register in vivo and ex vivo data. This is stuff that Lila Zolai did. Yeah, that's not supposed to say that. That's supposed to say Lila Zolai. And here we built some tools to register in vivo and ex vivo data. And this is hard because you can see the dramatic deformation that happens ex vivo. So when you do an autopsy and you take the brain out, you cut into the dura, the CSF spills out, kind of gravity takes over, things sag. There are these large scale deformations that occur in the tissue. But we would like to establish a pointwise correspondence. We would like to know that this point in the in vivo is exactly this point in the ex vivo. Right? And so one observation that we made, I should thank the group at UW who provided this data. This is from the ACT study, is that if you do a cortical surface reconstruction of the in vivo data and the ex vivo data, you can again kind of see the change in overall geometry. Like this one, the shape of this is pretty different than the shape of this. But the red-green patterns are the cortical folding patterns. So this is the central sulcus, the red are sulci, the green are gyri. And you can see that the folds are pretty much perfectly preserved. That is, here's like the omega, that's typically the human hand area. And you can see it like right there. Right? So there's this kind of large scale deformation that occurs, but the folding patterns are preserved. And so if we can align folds, then we can account for the vast majority of the nonlinear deformation that occurs across death. And actually, the truth is that this registration is much easier than the cross-subject registration, again, because the topology of the folds is preserved. And I'll talk about that in a minute. So here's an example. This is the in vivo scan, it's an MP-RAGE that we've already warped to be in register with the ex vivo. This is the 200 micron ex vivo, and I'll go back and forth. So you can see that they are pretty perfectly registered. Right? And this now lets us use this scan as kind of a neuropath magnifying glass. That is, I can look at the scan. This is Brian Edlow, who's a neurologist at MGH that I've worked with for years. And Brian looked at this 200 micron scan and he said, oh, look at the white matter here. This white matter is rarefied. Like if I had seen this on the in vivo scan, I would have recommended some downstream follow-up of it. And if we go back to the in vivo scan, we can see that it was there. Right? There's kind of this darkening of the white matter here. But it wasn't salient enough in vivo to be picked up by radiologists and neurologists at the time. And the same thing elsewhere. So here's a bit of white matter that's brighter than it should be. And again, if we go back to the in vivo scan, we can again see it was there, but it just wasn't seen at the time. It wasn't red. Okay, so now I'm going to talk about registering the ex vivo data. And here we're going from 1 to 3 micron light sheet fluorescent microscopy, or the 75 nanometer two photon, which we don't have much of yet, so we haven't really done anything on this, all the way out to the full brain or the whole hemisphere MRI. And as I said, this is a challenging problem. It's nonlinear. It's cross-contrast kind of by design because we want the different images to tell us different things about the brain, so they have to have different information contained in them. And it's wildly cross-scale. It's like 1 micron pixels to 120 micron pixels. So whatever that is, 100 cubed difference in voxel volume between these and those. And then the 1 millimeter, of course, are even coarser. Okay, the OCT is a critical interim target because it should exactly match the light sheet data. So the OCT, we take the image, we cut it, and then we do light sheet on that cut. And so the OCT image is exactly the same tissue as the light sheet data, but the light sheet is deformed by clearing, cutting, and mounting, this kind of thing. Okay, so as I mentioned before, there are cross-modal similarity metrics, things like mutual information and correlation ratio, but they're not great for nonlinear registration. You can get them to work, but they're kind of flaky, and it tends to be a little bit ill-posed. That is, you can generate nonlinear registrations that are the wrong one that will minimize those things or maximize them if you're trying to maximize them. And so we would like to kind of find a metric that's more robust than this. But of course, we already know how to do this because we've done it with the synthomorph shapes. Right? We've generated these synthetic networks that know how to do nonlinear cross-modal registration, and they've figured out their own similarity metric because all we told them was make sure the labels align. And they figure out what image intensities, what matching of image intensities result in label alignment. Okay, so here are some examples. This is Yu Wang's work. He was another postdoc that came from across the street. He did a PhD with Justin Solomon in kind of geometry processing. Here's an example of a calretinal stain. This is light sheet fluorescent microscopy. This is OCT of the same tissue. You can see why this is a hard registration problem. Right? There's a lot of deformation between these two. They have different contrast properties. There's all sorts of stuff going in the light sheet, all sorts of intensity artifacts in the light sheet data, these kind of exponential light attenuation and these areas where the tissue wasn't quite flat, and so we lose signal. And so for this, we don't build our image matching based on image intensities. That is, I'm not trying to say bright stuff here should align with bright stuff there. Instead, what I'm doing is segmenting the boundaries of the tissue and maybe the white matter, and then trying to align these segmentations. And this is how that looks. That's what the warp looks like. And this is the accuracy of it. So it works pretty well. So we can generate a pretty good alignment between the calretin stained light sheet image and the OCT. And kind of another amazing thing that you did is he derived a kind of general purpose Laplacian solver that is incredibly fast. And in general, when you use the Laplacian solver, you either use a direct solver where you give the problem setup and it gives you a solution that's not terribly accurate. But if you want accuracy, you have to use an iterative solver. And your iterative solvers basically recompute the solution until you achieve a desired accuracy. So if you say your tolerance is 10 to the -7th, it might run for 4 hours until it gets there. But you developed a direct solver that has the same accuracy as the iterative solvers. And the reason that's important is that for us, I mean, I think it's important for a lot of things. The reason it's important for us is it allows us to do this registration in real time. And that is, he's built a technique that estimates a time-varying velocity field that encodes the deformation. And if any of you do nonlinear registration, time-varying velocity fields take hours to integrate. Like to get a solution, it takes hours of this size images. But Yu's technique takes about a second. And this was the slide I was waiting for this morning, Emily. Because Yu was working on it till this morning. This is like a Python demo. We're gonna integrate this into our visualization environment. This is not integrated at all. But it shows that this technique can be interactive. And it's fast enough that I can take, so this is the source image, this is the target image. You can compute the initial warp and then iteratively improve it by picking point correspondences. So you can say this blood vessel is that blood vessel, and this spot in the brain is that spot in the brain, and it will iteratively recompute the registration to update the accuracy. So there's the initial registration, and I'll note the actual registration is faster than the movie playing. The registration takes about a second. Then you can pick correspondences. So you pick a point there and a point here, and it will update the warp. And then pick another couple points, and it will update the warp. And so we're going to integrate this into our visualization tool. And I think that for BICAN, this will provide most of what we need, because what we need is not to register 1,000 images an hour. We need to register kind of a handful of images accurately. Okay. Then from the, right, so now we can image the light sheet to the OCT. We registered the light sheet to the OCT. Now we want to register the OCT to the MRI. So here's an OCT scan, here's the MRI. The OCT is not very distorted. These are gentle nonlinear distortions, mainly because when we do the imaging, we're kind of physically distorting the tissue a little bit. We're putting it in a bag or a tube or something. And so there are residual nonlinear distortions between them. And this is Malta's work. This is quite old now. This he did like earlier in the week, I think Wednesday. And so he just took the synthmorph shapes that I showed you before, and he applied it to this pair. And it works pretty well. So here's the deformation that's induced. So again, it's pretty gentle. And here's an example of the accuracy. So I go back and forth. You can see that the MR and OCT line up pretty well. Right? OK, so this is where we are now. We can generate pretty good registration from single micron light sheet data all the way out to 100 micron ex vivo MRI. From the 100 micron ex vivo MRI, we can kind of go anywhere. I'll show you that in a little bit. And by anywhere, I mean like in vivo atlases. And the registration is pretty accurate, but it's accurate at the scale of a millimeter or so. Like if you look at it, you can see that there are inaccuracies, particularly in the interior of uniform regions. And that's understandable because we don't have a lot of information in the interior of the white matter. And we can't tell where the superior longitudinal fasciculus is or something. Right? We can only tell that it's kind of bright or dark or whatever. And so we would like to improve on these techniques to align at least mesoscale features, columns, layers, microcolumns, mini columns, this kind of thing. Okay? So how are we going to get there? Well, we know that vessels occur at almost every spatial scale in the brain. So down to the 4 micron capillary bed where a single red blood vessel can fit into a capillary. Vessels occur from there all the way out to the 4 or 5 millimeter peel vessels that run along the surface of the brain that neurosurgeons use to find or avoid motor cortex, for example. And so if we could segment the vasculature in this array of images, we could use vessels as endogenous fiducial markers. And we could say, I want the vessels to align across different image types, right? And of course, the vasculature is super interesting in its own right. As we were discussing at lunch, it's what fMRI is based on, knowing where the blood is, and lots of pathologies are vascular pathologies. Okay, yeah, so I already said this. So here's an example of an MRI to OCT registration. I think Adrian and Yale wrote this some time ago, and this is an example of why registering vessels is important. So I'll show on the left, this is a pure mutual information-based registration. So I'll go from the OCT to the MRI. And on the right, we trained a network with mutual information, but also telling it align vessels. And here we manually labeled a set of vessels in the source and target. And so we kind of, we encourage the network to find a registration that also aligns the vessels. And you can see that there are a lot of vessels around, but they're very small features. And so a general intensity-based metric won't align them unless you force it to. So here's, just go back and forth. And so, like here, you can see that that vessel doesn't line up unless, whoops, wrong way, unless you force it to. And if you force it to, then you get a similar registration, but now the interior of the stuff is lining up as well. And so if we could segment vessels in this array of imaging types, we could embed them in the registration and tell the registration, I want you to align stuff, but I really want you to align the vasculature. Okay? And so this has led us into a many years long hobby of segmenting vessels. And this was Yale's work initially, and now Chiara Mori and Etienne Gallet are engaged in it. And so as I mentioned, vessels occur in every spatial scale, and so they're present in every image we acquire, in every brain region. But they're super hard to manually annotate. And I said before, we owe years of therapy to our RAs who we forced to do manual annotation. We owe decades of therapy if you've been trying to manually label vessels. And it's because of their properties, because they occur at the edge of the resolution you can see in every imaging modality, unless you're getting like micron images. So in like 120 micron MR, there are 150 micron vessels that you're like, was that a vessel? I guess. In the 10 micron OCT, there are 5 micron vessels that we catch glimpses of. And so at every scale, it's really hard to see them. And they're everywhere. And this is my favorite quote from Yale, vessels are everywhere and nowhere. And that's because they occur in every bit of the brain, but they're only like less than 0.5% of the brain by volume, of the tissue volume. And so because they're so small by volume, they don't naturally line up using image-based intensity metrics for similarity for registration. Okay, so we decided we wanted to register them. We wanted to segment them to embed them in registration. I don't have, maybe on the next slide. And as I said, this is just a super hard task, and we've played this game so many times in the past couple of decades where we want to segment something, so we'll go out and manually label a bunch of them, and then we'll train an algorithm based on the manual annotations. And that completely failed for vessels. And the reason it failed for vessels is that the manual annotations just weren't good enough. And it's not because the manual annotators failed, it's because it's too freaking hard. And you can see that here. So this is an OCT. So this is an on-FOSS image. This is in depth. You can see the striations that occur by the exponential light attenuation. And so this was a super careful labeling. I think Caroline Mignon did this, and she's super good at this. She's so diligent and works, hours on these things. But even after working hours on these things, it's easy to find ones that are missed, here and here and here and here. And even this labeling of ones that I miss, like I missed ones, right? There's probably one there, one there, one there, one there. And the only way to tell that they're vessels, like these little dark spots, is to go through slices and say, well, does it look like it travels through slices? Is there a tubular structure in 3D that I can infer is present in the sequence of images? And that's really hard. Okay, so in OCT, it's really hard. Here's a similar example in ex vivo MR. Like, these bright white spots are vessels. The vessels can occur as bright spots in MR, but they can also occur as dark spots. So there was a dark vessel there. I'll play this movie again. You can see some diving when there was a diving vein there, probably. Here's one here. And it's just kind of random depending on whether there's any blood left in the tissue, because the blood has iron content and that makes it bright on this type of MR. But if there's no blood, it's just kind of an empty space and it looks dark. And so it's even harder because now you're looking for bright stuff or dark stuff. Okay, so what can we do about this? And the answer is kind of the same answer as before. That was a very slow transition. Is we can synthesize our way out of the problem. So we can generate completely synthetic vascular trees. And this was stuff that Yale did. Yale wrote this code where you start with kind of a root vessel, and then you randomly select like a caliber, tortuosity, a probability of branching, and then you let it branch. And you grow these vascular trees. Here's one with high density. So density is one parameter, and here's one with low density. And then Etienne and Chiara have been working on this since then. Etienne has been working on the OCT side, and Chiara's been working on the ex vivo MR. But the game we'll play is to take this synthetic vascular tree and build a synthetic image around it. This is what a synthetic image looks like for MR. There's a synthetic MR image, and then we'll embed our synthetic vessels in our synthetic MR image. So here's the synthetic image. These dark spots are the vessels. The red shows where the vessel location is. So it's the same image with and without the vessel labels. And now we know exactly where the vessels are. And we get the advantages of the whole synthesis game, which is that there is never a mismatch between the image and the labeling, because we construct the the image from the labeling. So the labeling is always exactly correct. But then the question is, do these images capture enough of the variability we see in real MR to generate a good vascular segmentation? And the answer is a qualified yes. So here's an example. This is a manual annotation. This is the other way. This is a manual annotation. This is the network, and this is the overlay. And so it works pretty well. It works pretty well for some cases, for some sequences, but it doesn't generalize. That is, if I tried a different kind of MR, it would fail. If I give it hip CT, it might fail. Okay. So, as I said, this works pretty well. We've gotten it working reasonably well on OCT. We've gotten it reasonably working well on some ex vivo MR, but there are a ton of things we would like to register. And so there are a ton of things we would like to be able to segment vessels in. The ex vivo MRI I showed you, there's a ton of in vivo MRI done in the vessels. Mostly the contrast is generated by moving blood, by the fact that you know that the blood is moving. This is kind of the feature that in vivo angiography uses to generate contrast in the vessels. But there are other kinds. In MP rages, we see vessels for reasons I won't get into. They're quite bright. In OCT, we see them. In light sheet, we see them in many different stains. We see them in the two photon and confocal, H&E. There's new hip CT data that shows the vasculature in just beautiful detail. I don't know if people have seen the hip CT coming from Claire Walsh and Peter Lee, but it's just lovely. And then, Jeff Lichtman is getting micro CT and EM that we also see the vasculature in. So we would like to segment them in all of these modalities. And if you work hard, and this is Chiara's work, and Chiara has done a great job kind of trying to tune the synthesis, both the vessel synthesis and the image synthesis, to work in different imaging modalities. but it's a pain. And you have to, each time you have to, and when a network fails, there's no easy way to know why it failed, like except just looking at the data. Like, well, I guess the vessels in my training set weren't large enough caliber. Like they didn't see these big vessels out here. So maybe I'll tweak my synthesis so that the vessels it sees are larger caliber, and then I'll wait 2 weeks and try it and hope my results get better. And that you could do that, and it'll work on time of flight images shown here. It'll work on XVMR and OCT. This is around the central sulcus. This is a different type of flight. This is a kind of more standard MRA, magnetic resonance angiography, slightly thicker resolution. I think this is a phase contrast. And then we got some hip CT from the London group, and it works. After a little bit of tuning, it works pretty well on hip CT also. But we would like not to do this. We would like not to have to play this game where for every time someone gives us an image, we have to figure out why our tools fail on it and what we can tweak in the image synthesis in order to improve it. Okay, so how can we solve this problem? How can we learn to segment vessels without handcrafting the synthesis for both the image type and the vessels? There are a giant number of parameters that go into the synthesis. Benjamin did an amazing job in the original SynthSeg paper at optimizing this, but he had, I don't know, 20, 25 degrees of freedom. You can't do a grid search in 25-dimensional space when every point takes 2 weeks to generate. So more generally, we would like to learn to modify the synthesis to optimize the accuracy of the segmentation on real data. And this is distinct from learning a synthesis that optimizes the accuracy of the segmentation on synthetic data. This is a very different problem, and I'll talk about why that's the case in a second. And then, of course, this would be super interesting because it would tell us something about how networks learn if we can optimize the synthesis. If we look at what the learned synthesis is doing, we could find out what's important for networks during training. And we always kind of had this idea, and it kind of underlay a lot of the synthesis research, that if you train a network in a difficult environment, it will grow up to be a happy network. And I don't mean that to be general parenting advice. If Danielle is watching or Regini, that doesn't mean anything about parenting. But this was kind of the basis of the synthesis stuff, is that we would show the network such difficult stuff that by the time it got to real data, it was easy. Okay? So can we show evidence of that? And I'll show you what I mean in a second. Okay, so this is work with Yale and with Jialing. This is kind of the normal synthesis framework. That is, I have a set of labels, call them Z. I stick them through my synthesis augmentation engine that generates an image I'll call X, a synthetic image X. I'll stuff that through my segmentation network. It generates a prediction Y. And then I put it in a loss. And the loss compares the ground truth labels to the predicted labels and says, well, they don't overlap that well. And so I update my network parameters to make the segmentation more accurate on the synthetic data. Okay? And then the general optimization procedure we go through is messing around with the parameters of this thing and hoping that it'll get better on real data. So what we did is embed this optimization into the entire network training. And this is a difficult problem because, so here I have a segmentation network. This is the same segmentation network, so I just copy the weights from this down here, and then I can apply it to real data. Right? So I can take a real data set, I can stuff it through the segmentation network, I get a real prediction, I compare it to some ground truth labels I have, but I can't optimize this with respect to the synthesis because the synthesis isn't in this loop. That is, if I change the synthesis, it doesn't change the output here. So the gradient of this with respect to the synthesis is 0. There is no gradient flow that goes back to the synthesis engine. But what Yale's kind of aha moment was that, and I don't need to go into details of this, but basically the change in this with respect to the change in the weights is not 0. And so if you keep around the weights from kind of the last gradient step, you can flow the gradients back from real loss into the synthesis. And now I can build a learnable residual network that is an image I add to every synthetic image that optimizes not the synthetic loss, but the real loss. And critically, these real labels never go into this network. So this network has no ability to learn from the real labels. All it's learning from is what to do to the synthetic data to optimize the accuracy on the real data. Okay? And the reason I say that this is very different than optimizing the accuracy on the synthetic data, because optimizing on the accuracy on the synthetic data would be easy. Right? I just make it a binary image where my vessels are 1 and everything else is 0. That's not what I want. Okay, so this works astonishingly well, at least in early days. This is again about a week old. But if we take kind of a standard synthesis and we train a segmentation network, we get a dice accuracy. So this is overlap here between the automated and the predicted segmentations. Without the learn to synth stuff, we get an accuracy of around 53, 54. And this is a little bit of a straw man because I didn't tune this at all. I didn't add noise to it. I didn't do a lot of augmentation. I wanted that all in the synthesis network. And it also, I just kind of randomly, I should be pointing these out and I haven't been. We have 7 64 cubes that have been manually annotated, mostly by Caroline Mignon, but also by our interns. Thank you, interns, for this thankless task. And what we found on the manual annotation side is that the only thing we can really do is segment very small blocks. That is what I showed you earlier was like a 300 cube. We can't do 300 cubes. There are too many vessels in it. And so we do either 32 cubed or 64 cubed. And that's small enough that you can go through it exhaustively, kind of time after time, and try and find these tubular structures that are in it. And I think I happened to pick 2 that are pretty difficult for the ground truth, for the kind of standard segmentation network without the learn to synth. So what does it look like? These are what the results look like. So this is the manual annotation. And again, you can see, kind of hard in the lights maybe, like this should be 1 vessel. But because the images are noisy, this is what the images look like. There are breaks in it in the manual annotation because the manual annotator kind of lost it for a little bit and then found it again. This is what the initial segmentation looks like, and this is what it looks like without learn to synth. With learn to synth. And so what is the learn to synth doing? If I go back here to the diagram, I'm essentially learning an image that I add to the initial synthetic image that I'm going to give to the network. Right? So what is the image that I'm adding, the initial synthetic image that makes the network more accurate on real data? And this is pretty interesting. So here's what the initial image looked like. So this is the initial synthetic image with the initial labels shown in blue. Kind of hard to see the blue here. This is it without the labels. So this is the same image. So these are vessels here. This is just noise. These are synthetic structures that are not vessels. The learned residual is like this. I'll go back and forth. And so what it looks like is like a photographic negative. That is, what it's learning to do is make life hard for the segmentation network. And so if I add them together, this is now the input to the segmentation network. This very low contrast image. And so again, this is early days. This is a result that's like a week old, and I don't know that it'll hold up. But it seems like the answer is make life really hard for your networks during training, and then your inference will get better. And there are a lot of things to be done in this. Like we very much want to add parametric variation. So for example, vessel caliber should be a learnable parameter. What is the range of vessel calibers that optimizes the accuracy on real segmentation? Because for example, in the HIP-CT, we see all of the vessels. We see vessels from like the 4mm peel ones. Actually, probably we've taken the peel surface off, so we don't see that. But millimeter scale vessels down to 10 micron vessels. But in the OCT or the exterior MRI, we don't see the 10 micron vessels because our image resolution is 120. So I think there's still many games to be played. And this is a quantification of, so this is the learning, this is training across time, so epoch, and this is negative accuracy. So down is better here for no very good reason, except that I made the slide like 2 days ago. And so this is the accuracy on the synthetic data, and this is the accuracy on the real data. And what you can see is that the network learns to improve the accuracy on the real data about the time it learns to decrease the accuracy on the synthetic data. So it's kind of some quantification of what I mentioned before. Okay, that's it on the vessel segmentation stuff. Questions or? Yeah.
Person to person.
Oh man, I wish I knew the answer to that question. I think that's funny. There was when I was a postdoc at MGH, there was a postdoc named Rick Hogue who's now at Montreal, and he was very interested in vascular imaging, and he was like, we should build an atlas of like cerebral vasculature and see how it varies from person to person. And I was like, that's a great idea. That was 30 years ago, and we haven't done it. And there are lots of basic unanswered questions about the vasculature. So, for example, do the vessels follow the fascicles? If we could segment the vessels, would we know where the major fascicles are in the brain? Maybe. I mean, you expect they're supplying perfusion. Like, it might be that if we had a vascular map, we wouldn't need diffusion MRI. We could just look at the vessels and know where the major tracts are. I don't know. Do they delineate cortical areas? Divernois did these India ink stains that seem to show lots of vascular change in some regions. A lot of it was V1, V2, and of course, V1 changes along every dimension from every other cortical area. But it may be that the vasculature tells us something about cortical areas, like where are the boundaries between this and that cortical area. So the answer is, I don't know. Like, I would love to know the answer to that question, but I don't know. Okay.
Another question.
Yeah.
You mentioned these white matter hyperintensities.
Mm-hmm.
I guess those are still kind of like.
Oh, sorry.
Oh, yeah. You mentioned these white matter hyperintensities, which are still, as far as I understand, kind of a mystery object. And one of the theories is that they're vascular and that there's some kind of vascular defect. So just curious what you think.
Yeah, let me start by saying I am absolutely 100% not an expert on this. But I have spent time close to some experts, so I know a little bit just from eavesdropping. So I think that there are different kinds. When we look at these lesions on MR, sometimes they're like a little, on a T1 weighted MR, they look a little darker or a little brighter than on a T2 or a flare or something. But sometimes they're like black holes. And I think those probably have different origins. I think the black holes are probably like dead tissue, whereas the other ones may be, some inflammation. I also think that they change in different, so like in MS we see them, and that probably is different than the ones we see in aging and Alzheimer's. But in the end, I don't know that we know the answer to that. One of the things that Dirk Keen was excited about when we first started this whole project is we can find them with MR. Like for the neuropath, they're hard to study because you don't know where they are. Like you don't see them on a slab, but we see them in MR, and then we can project that onto the slab, and then you can go and take that out and look at it. And in fact, we did that with the case I showed you. It's just the neuropath wasn't very interesting in that case, so we don't typically show the neuropath findings for that. But the techniques are all in place to do that, like use the MR as a spotlight to tell the neuropathologist something's going on here. Can you check it out?
You could find a slab with one and then go in and dissect it straight to it.
Yeah. I mean, the one caveat is that we typically do the best that we can do. We typically need some type of image of the whole brain, whether it's photos of every slab or an MR before it was taken out, or a 3D surface scan. Like once you remove parts of the brain, we can no longer recover the topology of it. And topology is a pretty strong constraint. So that helps us a lot. Okay, on to individual level stuff. Okay, so let me go back to this. So here we would like to increase the level of detail. So for years we've done anatomical modeling of caudate and putamen and hippocampus and amygdala, and we've extended that to things like subfields in the hippocampus and amygdala nuclei. But we would like to extend it in individual subjects that we're doing this type of high-res imaging on, so that we're not just saying it's in the frontal lobe, but we're saying it's in layer 3 of Brodmann area 44, for example. And so we've been trying to extend the scope and level of detail of our models to kind of incorporate these mesoscale anatomical features into them. And this is work that Taylor Zhang has done, where we've done some manual labeling. I will again thank interns, because interns were involved in manual labeling here. So the game we play here is we image the entire hemisphere at 120 micron. We project our in vivo estimate of where area 44, 45 is, Broca's area. I'll explain how I do that in a little bit. And then we take a block out, we do the OCT, we do the light sheet imaging. And so we would like to start analyzing not just the gray matter as a whole, but layers within the cortex. And on the ex vivo MR, I don't know if you can appreciate it here, we see the boundary between infragranular and supragranular cortex almost everywhere. It's a pretty salient feature. There's theories about how they developed separately and came together and this and that. But for us, it's a feature we can see across the whole brain, and we can manually annotate it. And so we've done, let's see, how many? So this was 15, I think we're up to about 20 now, where we have hemispheres manually annotated, infragranular and supragranular cortex in Broca's area. We also have 2 hemis where we've done kind of a scattering of slices elsewhere. And this is a huge amount of data, 120 micron across the hemisphere. At 1 millimeter, we get something like 1 million voxels in the brain. So at 120 micron, we would get like 1,000 times that, I guess. So it's a lot of data, and it's not feasible to manually annotate the whole thing. And so we've been trying to develop techniques, and by we, I completely mean Taylor, whose idea this was, techniques that use manual labels where they're available, so supervised training, but also benefit from the rest of the data set where there aren't any labels. Right, and Taylor estimated that it would take like 20 years to take our existing personnel to label every slice in every brain. And none of us are that patient. Okay, this is the next slide. Yes, okay, so the way this works, so Taylor developed a technique that's semi-supervised. That is, it uses the labels in the labeled regions, but it uses the unlabeled data as well. And the way it works is this thing called cross-pseudo supervision, which is one of these deep learning things that I would never have thought would work. And the game he plays is that instead of training a single network, you train like a little sibling group of networks, like 3 of them at the same time. And you sample a voxel from the brain, and you say, is that voxel a labeled voxel? If it's a labeled voxel, I'll train all my networks on the label. If the networks make a mistake, I'll penalize them. But if they make the same mistake, I'll really penalize them. Like, so it's what he calls an error diversity term. That is, he wants the networks not to become one network. He wants them to learn different things. Then if he samples outside of the labeled region, he has each network train the other. And so there's no ground truth out there. And I would have thought this thing would converge to nothing very interesting, but in fact, that's not true. In fact, it works. Here's an example of a whole hemisphere segmentation. Here's the quantification of it. So these are different cortical areas. These are our estimates of what Brodmann area we're in. This is entorhinal, this is V1 and V2. And you don't have to look at all of these, but the interesting ones are this. So the other thing I'll add is that this is a multi-resolution UNet cascade. So we give the whole data set at low resolution to the first UNet, and then the second UNet looks at twofold upsampling and also gets the whole brain context, and the next one is another twofold upsampling until we get to the original data resolution. And so the last UNet has the benefit of the whole brain context plus the highest resolution we get to. Anyway, so that's shown here in purple. NNUNet is kind of a popular baseline that people use. And so you can see that most of our labels are in 44, 45, and they all do pretty well there. You know, 44, 45 has its own architecture. It's 5-layer cortex. There's no layer 4. It's very granular. And so if you take this network trained there and run it in V1 or V2, it completely fails because it's an architecture it's never seen. It's a cortical architecture that's just different enough from the training set. But the cross-pseudo supervision keeps this accuracy pretty high. And so we now have tools that in the ex vivo 120 micron ex vivo MR do a very accurate job at labeling infragranular and supergranular. And probably we're not going to do a lot more, not clear, but we don't see 6 layers many places at 120 micron. We see it some places, but we see it everywhere in the light sheet data. And Patrick Hoff and the group in Lens, Irani Costantini, are generating manual labelings of all 6 cortical layers that we're going to look at. But so how much use is this if we can generate accurate infragranular, supergranular segmentations on 120 micron ex vivo data when we're not going to get 120 microns in vivo anytime soon? You know, if we struggle like crazy, we can get, I don't know, 400 micron. Like, that's a long scan. It's very sensitive to motion. It's very difficult to get beyond that resolution in vivo. And even 400 microns is really pushing it. And so what we've been doing, and this is Ola's work, is we've been looking at how accurately we can predict these boundaries in the absence of imaging data. That is, if I can tell you at this point in the brain, the layer 4 boundary is always 1.3 millimeters from the gray-white boundary, I don't need to see the contrast. I know where the boundary is. And so here's an example. I'll go through the details of what this means. So let's see which is which. The ground truth is yellow. So the yellow is the one that was generated by the manual labeling. And then the green and the red are two different different models of cortical folding formation. One is called iso volume, one is called iso distance. And essentially they're used to estimate the orientation of columns and how far out the cortex, the different boundaries are from each other. And they both fit reasonably well. Probably the iso volume, if we just do a single parameter of the whole brain, iso volume is probably a little bit better. But if we do a vertex-wise fit, that is, if every point in the brain, we fit an iso volume and an iso distance model, that's what's shown here. This is an error map. This is the distribution of errors. They're about the same. So vertex-wise, you know, if we use our 17-ish subjects to fit the models at every point in the brain, we can do a pretty good job at predicting the infragranular and supragranular border, and whether we use an isodistance or an isovolume doesn't really matter very much. But more importantly, if I fit a full linear model just based on geometry, so T here is the thickness of the cortex, K1 and K2 are the principal curvatures. And so here, this is a linear model. I want to recover these betas. At every point in the brain, we can do an excellent job at predicting where the infragranular border is. So, what is this? Like the mode is around 1/10 of a millimeter. And this, so if we can generate accurate surface models, we can predict where the boundary is, even if we can't see any contrast that separates supragranular from infragranular.
We have a question online, Bruce, if you don't mind. So the question is, can the approach your group developed for training on synthetic data for brain mapping be applied to other organs like the heart? And what challenges might arise in translating these methods to other systems?
The answer is yes. I mean, so the shape stuff is not organ specific. It's completely synthetic. Cardiac deformations are a lot bigger than cross-subject variability in the brain. So I expect there's some tuning that would have to be done. I think Chiara has applied the vessel stuff to non-brain data. I think she applied it to some kidney data, I think. I'm not sure. So in principle, yes. In practice, we haven't done a lot of it because we're so brain-centric. Okay, so we have put this whole thing together, at least on the individual subject basis, where we've acquired light sheet data, we've acquired stain data, we've acquired OCT, we've acquired MR, we've registered the whole thing, we put it into the same coordinate system, and now I'll show you a fairly gratuitous fly through so that's the MR, that's 120 micron MR, that's the 55 micron MR from the block, this is the 3 micron OCT, and then we zoom into initial stain in motor cortex, where we can see, every neuron in motor cortex. And then we could start to quantify this stuff. Oops, don't need to play it again. We can start to quantify this stuff. So the stereology was done by Patrick Hoff. The light sheet was done at Lens. The OCT was acquired locally, probably by David Buzz, but possibly by Hui Wang. But we can project this light sheet data through the OCT. So here's the light sheet data through the registered OCT, or rather the registered light sheet data to the OCT and then back out to the MR. We have cortical surface models of different layers in the cortex, and we could start to make maps. So this heat map is basically stereology at different layers on the cortical surface. So this is all the way out the peel surface. So think layer 1, and then down here to layer 6. And so we can count, I think this is new N. Yeah, so this is like all neurons. So how many neurons are there in layer 1 of this bit of the cortex in a human brain? Okay, almost at the end. Any questions on that? No. Okay, so now I'm going to talk about individual subject stuff. So there are a series of papers published in the late 90s, early 2000s by, this is actual Schleicher's paper, but mostly by Katrina Muntz and the late great Carl Zillis, who was an amazing neuroanatomist who died a few years ago. And they developed this technique they called observer-independent 2D histology. So this is a photomicrograph of a Nissl stain. Every dark spot is a neuron. They fit the surface, the gray-white surface, the peel surface. They solve the Laplace equation that gives them a streamline connecting the two. And then they built what they called a GLI, which is a gray level index. And basically it says how many neurons are there at this spot in the brain. And they built a technique that looked at changes in this GLI. So these peaks are changes in the GLI that represent potential boundaries in the data. So where's the V1, V2 border? Where's the V2, V3 border? This kind of thing. They published a series of quite influential papers in the 90s looking at this in various cortical areas, and they used it to try and characterize human variability. And it was a set of papers. So these are, I guess, 2 of Katrin's papers looking at Robin area 17, 18, 44, and 45. And generally the conclusion was that the microstructure is variable in ways we don't have access to using macrostructure. That is, we can't use macrostructure to predict microstructure boundaries. And they quantified this. So this is V1. This is a heat map. So red means they have 10 neurologically normal subjects that come to autopsy. They do unbelievable amounts of work to generate this stuff. Red means all 10 subjects agreed that this spot was V1. And yellow is whatever this is, 6 subjects agreed. So even in V1, which is the most architectonically stable feature of the brain, it's always in the Cochrane sulcus. It has one of the few architectonic features we can see with the naked eye, the sort of Gennari. Even in V1, there's this tiny little dot in the middle where all 10 subjects agree in this coordinate system. So this TEDARC is was a standard coordinate system in neuroimaging. It's less so now. Anyway, even in V1, in this coordinate system, the V1s don't line up. Like my 10 subjects don't overlap that much. And I go one step up in the cortical hierarchy and I don't have any red. Like the majority of my distribution is like 4 or 5 subjects. And then I get out to language cortex, 44 and 45, and this blue means no 2 subjects agreed. Like every spot was in a different cortical area in a different subject. So Katrin was kind enough to give us her data. We do registration differently in the brain. We don't use image intensities, we use geometry. So this is kind of an outline of what we do. We have surface models of each subject. This is 4 subjects. We map these surface models onto the sphere. So these red-green patterns, red is the central sulcus. The red are folded in, the green are folded out. And then the game we'll play is to extract summary statistics across subjects and shift these red-green patterns around on the surface of the sphere until they line up, so that I get a diffeomorphic transform with each of these. I can go out to here and then back out to any individual subject. And it's a coordinate system that kind of maps, it normalizes out the variability of anatomy we see in cortical folding patterns. Okay? So this is fairly old at this point. I don't have a citation. I don't have a citation. We published this, I don't know, 10 years ago or so, where we got Katrin's data. We did the exact same thing. I've changed color scales. I'm sorry. So this means 100% is all 10 subjects agreed, and gray means no 2 subjects agreed. We did the same study. We just analyzed the data, mapped it to surfaces, registered the surfaces, and counted how often the subjects agree. This is V1 or V2 or whatever. And what you can see is that in V1, cortical geometry is a perfect predictor for the architectonic boundary of V1, V2. And this isn't surprising, like we know V1 is in the calculus. In V2, it's still quite good. And even if I get out to 44 and 45, there's some variability here, but there's this large core of the area where we're 100% certain we're in this architectonic region. And so we have some confidence that we can predict architectonic identity just from geometry. We replicated this. So this is a photomicrograph of a Nissl stain. So this is the PL surface. This is the white matter boundary in a control subject. This is an Alzheimer's subject in entorhinal cortex. Entorhinal is devastated in early AD. This is like the earliest clinically diagnosable form of AD, and 60% of these neurons are dead in layer 2 of entorhinal. We went looking for entorhinal cortex with ex vivo MR. This is 100 micron ex vivo MR in the medial temporal lobe. And you can see, oh, sorry, I should mention, go back to this. The entorhinal has this bizarre architecture, these entorhinal islands. They're cell dense clumps, and then like a 250 micron cell pore area, and then another cell dense clump. As far as I know, this architecture doesn't occur anywhere else in the human brain. We went looking for them with ex vivo MR. Here they are in ex vivo MR, these little bright spots. And this let us find the boundaries and say, are they predictable with MR? And the answer is yes, we can predict where entorhinal cortex is. And further, we can take this data and map it to Alzheimer's data. This is data Randy Buckner gave me. And we can predict where entorhinal cortex is and see if there's thinning. And so this is the thickness of the cortex of controls. This is the thickness in Alzheimer's subjects. And so you can already see there's dramatic reduction in cortical thickness in these very mild Alzheimer's subjects. And if I take this ROI and I translate it just to posterior, a centimeter posterior, this effect goes away. So it's not widespread thinning across the whole brain. It's actually in this predicted architectonic region. I'll skip this. Okay, so we would like to establish a common coordinate framework that does cross-optical alignment of some relevant properties. What properties? We would like to do it even if we can't directly observe them at inference time. Geodesics I won't talk about. And then we would like to be able to extract summary statistics. What's the average human look like in this coordinate system? So what properties would we like to align? Well, there are a lot of them. We'd like to align site architecture and myeloarchitecture and vasoarchitecture and the vasculature and molecular properties and geometry, et cetera. But we don't even know if this is possible. We have no idea if these things line up with each other. So how can we improve on what we've already done? Well, we tried this years ago, Paulina and I, with Thomas Yeo, who's a grad student who's now back in Singapore, and Mert Subanshu, who's in at Cornell Tech. And the idea was that maybe all folds aren't equally predictive of all areas. I mean, it's not a deep idea. Like we know that calculin is predictive of V1, but can we build a computational framework that'll let us extract this information? And so the idea here, and this is Andrew Lee's work, is can we train a deep network that is given a map of cortical folding patterns? And you tell the network, I want you to align the folding patterns. Like this is what the network sees. These are the things that go through the convolutions, but I also want you to align this other thing. And I'm not going to show you the other thing. I'm just going to tell you how well you did. Right? So think, I'm going to use fMRI as an example, but it could be anything. It could be a map of Brodmann areas. It could be, some proteomic image, whatever. So you give it the folding patterns, it generates an alignment, you apply that alignment to some other pair of images, and then you tell the network, no, your thing sucked, do it again. And so the only way it can solve this problem is by building a predictive model of the thing it doesn't see from the thing it does see. Right? So it has to learn to predict this unseen feature set from the feature set it sees. Yeah, maybe I'll skip this one. I'll just add that we also learn an atlas at the same time. So we don't register to an atlas, but we extract an atlas from the data set of both the unseen feature set and the seen feature set over the course of learning. And this is an example. So this is stuff from Evelina Fedorenko and Greta, who defended like 2 hours ago. So I hope it went well for her. I didn't get to go to her defense because it was today. And so this is human language fMRI. And human language fMRI studies have suffered from the fact that functional areas in the language networks don't line up across subjects for a long time. That is, I do an fMRI study of a bunch of subjects, I get robust activation in every one, I line them up in some common coordinate system, and I get nothing. And so this has made life difficult. Here's an example. So you usually get these like 3 areas in frontal cortex, and then we get this broad swath temporally. This is an average that we made with kind of our standard registration tools. And you can see that despite these robust activations, I don't see anything there. But what's interesting is if I look at every one of these subjects, there's like a little divot here. It's actually not a divot, it's a fold out. So this is the pre-central sulcus, this is the middle frontal gyrus, and there's this little interruption here in like every subject that has a functional activation when you do a language task, right? But we lose that because it's a small feature, it's variable, it doesn't line up. And so when we do geometry-based alignment, we lose that feature and it doesn't line up across subjects. And so the underlying functional activation that was there doesn't line up. But we can learn to line it up. Whoops, one too many. And so this is kind of our standard registration. This is with this learned atlas and learned registration. And you can see it recovers these kind of 3 spots quite nicely. And so this works for fMRI, but it could work for any unseen feature set. Okay, and that's all. I tried to mention people as I went through. A lot of people contributed to a lot of this. I also wanted to say thank you to our interns. So this was our fall 2024 cohort from lots of places in the area. This was the spring 2024 one. This was 2023 and 2021. We did it in 2020, which was a crazy time, as you might imagine, and we don't have pictures from that. And then I also wanted to thank Ren and Regini, who are the ones that run the internship, Julie, who's co-PI with me of the R25 that funds a lot of it, and Allison, who basically makes everything happen. As I think you guys know, we have a partnership with My Brother's Keeper, which is down the street, and they're the ones that really helped us do this. And then Sarah, who was involved early on, is still involved and enormous help, and Ted across the street. And then I wanted to thank Kiku, who, if you don't know, was chair of the PEDP working group, which is a typically thankless task. Anyway, so thank you, Kiku, for doing that. I know that it doesn't help your career in any way, so it was a kind thing to do. And that's all. Thank you. Any other questions now that we've come to the end? Yeah.
I'm curious to know if you've identified any spatial variations in gene expression that kind of help your models.
Oh man, you guys have to tell us that. No, so we don't have that data yet. I mean, I think Alan is trying to get that data. But no, we have we've been working on the kind of glue infrastructure for this stuff. And the glue is just starting to dry. I'm going to take that metaphor too far. But so no, we haven't applied it. I don't think there's much data on that yet.
But what would be your best bet on this imaging phenotype could be linked tightly to the spatial or any type of gene expression?
Yeah, I don't know. It's an interesting question. I know that I remember giving a talk. So I wish Randy Buckner was here. I remember giving a talk once showing like how we can parse like frontal cortex. And Randy's response was, I don't think there are any areas in frontal cortex. It's just like a broad, slow gradient. Like there are no sharp boundaries. And maybe he's right. I don't know. Randy's not wrong very often. But maybe he's wrong. Like we just don't know. We don't have that data. And so, it is astonishing to me that here we're almost in 2025 and there's this stuff we don't know. Like we segment Brodmann areas, 44, 45. Is it really a cortical area? Does it have different functional properties? You know, most of that stuff is from like vascular lesion studies done in the 1800s, or we just don't know. And so one of the things that I think is exciting about BICAN is this real push to start to build the infrastructure needed to answer these basic questions that have such implications for, you know, how we treat diseases, you know, how the normal human brain works. And I wish I had an answer to that one, but I don't. Yeah.
So you mentioned this really interesting work you guys are doing in that project of like functional imaging near the end of life,
Mhm.
and then kind of having that functional imaging guide the dissection of the
Yep.
samples.
Yep.
So I guess I had two questions about that. One is are you guys doing any other measurements near the end of life, like cognitive measurements or things like that, that could then be correlated to the
Yeah,
functional data?
that's a good question. My guess is not very much. I mean, these are very sick people.
Yeah.
And so it's not clear they can tolerate the imaging. You know, even the imaging itself, you know, we're very parsimonious with what data we're going to try and get because they're not going to be able to tolerate very much. So there may be some of that going on, but I don't know. So that would be a question for people at UW.
And have you guys as a team yet been able to test the hypothesis that the imaging guided dissection gives you sort of like more similar molecular results than if you had just sort of done it naively?
Definitely not. I'm trying to think how we would even do that. It's hard because we don't know what the answer is we don't even know what we're looking for.
Yeah. I guess you could just have a simple metric that like, if you really nailed the registration, then things would be more similar across people than if you didn't, right?
Unless they're not. I mean, I know Katrina Muntz always used to tell us that the variation in architecture across areas is much less than the variation in architecture across people. And I really hope that's not true. Like if that's true, it's big trouble for us. But maybe it's true. I don't know. I mean, there's just, you know, in neuroimaging in general, we always think of cross-sectional variability as the single largest source of variance. And that kind of drives the way we do analysis because of the variability. But anyway, the answer is some variance of no. So no, we haven't done it. But that data is going to be amazing if we can get it. You know, that will be super interesting. I mean, the other question is how we sample. Like, you know, we've had tons of discussions on how we sample. And that depends on what the underlying hypothesis is. You know, what are you trying to test? And there are different ones we'd like to test, and we're not going to be able to test them all.
What are some of the imaging phenotypes that show the most variation across people that's not just predicted by like age or something like that?
Oh, cortical folds, I guess would be the first one. Like folds are hugely variable for reasons we don't understand. But even things like, you know, ventricular volume, like you look at a subject with large ventricles, you know, some people without AD have large ventricles, some people with AD have small ventricles. I mean, it's one of the reasons why radiologists use change as their measure of choice, because then the kind of baseline values fall out, right? You don't care if the ventricle was big or small in an absolute sense, you just care that it's getting bigger or smaller. Yeah, but you know, in fMRI, there are people that are good activators and people that aren't good activators. What does that mean? I don't know. But repeatedly, I mean, Nancy Kamwisher earlier in her career was getting imaged all the time because she was a great subject. I don't know, maybe she was just really dedicated and didn't move, but maybe there's something about her vascular anatomy that lends itself to being imaged well. I don't know.
All right, I think we're all set then. Thank you so much, Bruce, for a wonderful talk,
Okay.
and thanks everyone for coming.
Thank you, it was fun.